last class, we actually learned about the biosynthesis of fatty acid, and then we focused on how these fatty acids are utilized to synthesize the different lipids, such as triacylglycerol, which are the storage form, and the other one, how they are incorporated into plasma membrane. Okay, so there is another lipid which is also very important, that is called cholesterol. Actually, cholesterol is present in animals. Okay, they are not present in plants, so they are actually important for animals, even for humans. But it is not necessary for us to get it from our diet. Okay, so that means we need it, but we don't need it from our diet because it can be synthesized in our cells and the precursors which are present. Now, because cholesterol that has a very complex structure, and it is made up of 27 carbons. Actually, all the carbons of cholesterol that actually comes from very simple precursor, and that simple precursor is the 2-carbon compound acetate. Okay, acetate, or we can simply say acetyl CoA. Acetyl CoA is the simple precursor of cholesterol, and we also learned that that is the precursor of fatty acid as well. Okay, now in the biosynthesis pathway, there is an intermediate produced which is called isoprene, and that intermediate is actually produced during the biosynthesis of cholesterol, as well as that also function as precursors for other lipids. So if you look at this here, there are the structures. So acetate, and if you look at the structure of cholesterol, so you can see especially the carbon which is colored in pink. So all the carbons here, they are coming from this simple precursor acetate, and the intermediate is also shown, isoprene, that is the intermediate which is also actually functioning as precursors for other lipids. And cholesterol, which is actually why we need it, this is very important for us, because if you look at this one, cholesterol which is required to synthesize steroid hormones, bile acids, vitamin D, and all of them, they have important functions. That means in our physiology, especially in our digestion system, as well as other physiological function. Okay, so next one is the biosynthesis of cholesterol. Here actually we are going to learn very brief biosynthesis steps, not elaborately. So again, the cholesterol is synthesized from the simple precursor acetyl CoA. Acetyl CoA is also the precursor of the fatty acid, but the pathway is totally different. And the major 4 stages are shown here. If you look at the very first step, where acetate, that is like coming from the acetyl CoA, so 3 molecules of acetate, they are condensed together to give rise to 6-carbon intermediate, which is called mevalonate. Okay, mevalonate. And in the next step, mevalonate is activated. So mevalonate has 6 carbons, and when it is activated, 1 carbon is released, and that give rise to the isoprene. So it is activated by ATP. If you look at the structure, 2 phosphate groups are added, and that is coming from ATP, which is not shown here. So activation is done by ATP. So that is the isoprene. Now, in the next step, the activated isoprene, actually 6 molecules of 5-carbon isoprenes, they are polymerized. That means one by one, they are added to each other and give rise to 30 molecule, linear molecule, which is called squalene. Okay, they are still linear. after the polymerization reaction, they give rise to a linear molecule, and that is called squalene, and squalene has 30 carbons. In the next step, that is the final step, we can say, where this linear squalene undergoes cyclization and give rise to the ring structure, the four ring structures of the steroid nucleus. So we can say the cholesterol, actually they are steroid compounds in nature, and all the steroids, they have ring structures, and the ring structure is known as the steroid nucleus. So there are four rings. So if you look at the structure, 1, 2, 3, 4, and also a like hydrocarbon chain is attached to the ring structure. And obviously maybe there are some changes occur according to the particular steroid, oxidation, removal, or migration of methyl groups, and ultimately give rise to the cholesterol. And actually that we have learned during the macromolecule lipid, where we the cholesterol is actually part of the plasma membrane, so they are membrane lipid, and like the other membrane lipids, they have the same feature, that is they are amphiphilic molecule. They have the hydrophobic part as well as the hydrophilic part. So in the structure here you can see the hydrophobic part is the ring structure, as well as the like aliphatic chain, and the hydroxyl group, that is actually representing the hydrophilic part. Okay, so that is about cholesterol. And here the reaction, actually if you want to learn the reaction by chemical structure, that is fine, but if you don't, that is even okay. Okay, now as we said that steroid or the particularly cholesterol is very important for us, because the important steroid hormones, they are synthesized from cholesterol. And what are the steroid hormones we are going to see? The first one, there are two steroid hormones which are actually synthesized in the cortex of the adrenal gland. Okay, so do we know where these adrenal gland are present in our body?
Ma'am, kidney.
Yes. So actually, here we are focusing on this in very briefly, but in course 2 or 3, you are going to learn about these hormones elaborately. So these gland are present on the top of the kidneys, and there are a part which is called the cortex of the gland, and within the cortex these steroid hormones are synthesized. Okay, the first one is called mineralocorticoids. So if you look at the name mineralocorticoids, minerals are involved here. So the function of this hormone is to control the reabsorption of inorganic ions, or we can say minerals, which are sodium, chloride, and bicarbonate, and that is done by the kidney. And second one is the glucocorticoids. So here, if you look at the name glucocorticoids, that is actually responsible in regulating gluconeogenesis. We have already learned gluconeogenesis, and also it has another important function in our immune system, that reduce the inflammatory response. Okay, there are other sex hormones, or steroid hormones, which in general we call sex hormones. They are produced in the male and female gonads and the placenta, and those are the sex hormones are progesterone. Progesterones regulate female reproductive cycle, and androsterons, example testosterone, and estrogens, example estradiol. These hormones, they are actually influence the development of secondary sexual characteristics in male and females respectively. That means testosterone is functioning in males and estradiol is functioning in female. So these steroid hormones, they are very effective, but we need a very low concentration of this hormone. So that means if we synthesize in small quantities, that is still enough to carry out the functions done by these steroid hormones. So that's why actually the required quantity, which is functional for the important physiological activities of our body, and we can synthesize the hormone by ourselves from cholesterol. So we don't need to take cholesterol from outside. Okay, so this is the pathway by which the different steroid hormones are synthesized. And if you look at this cholesterol, so from cholesterol, at first it is converted to pregnenolone, and that is converted to progesterone. Actually, from the progesterone, all the other steroid hormones are synthesized. So actually, that means this pathway is occurring in the cortex of the adrenal gland, as well as the male and female gonads and placenta. So we get progesterone, then from progesterone, cortisol, corticosterone, as well as testosterone, and eventually estradiol are synthesized, and the functions are also mentioned here, which is also written in the previous slide. Okay, so here the particular biosynthesis is shown here, but it is actually not so important at this stage, and this is also actually very complex pathway. But we can see that the precursor here is the cholesterol, and then the cholesterol is actually undergoes removal of the side chains, that means specifically the hydrocarbon chains, and that these biosynthesis reactions occur within the mitochondria. Okay, mitochondria of steroidogenic tissues, that means the adrenal cortex or the gonads. So, and the most important thing we have to keep in mind that the reaction which is happening, that eventually causes the removal of the side chain by the cleavage, or which occurs in between the carbon number 20 and 22. And another important thing that we must keep in mind, the reaction here, which is actually providing the electrons, and the electrons are eventually doing the cleavage of the bond, and that electron carrier here, that is called cytochrome P450. Okay, so cytochrome P450, that is has the function of mixed function oxidase. Okay, and the electrons um, delivered, the electrons are delivered by the NADPH, because this is also a biosynthesis pathway, and we already learned in the biosynthesis pathway, reducing power is provided by the cofactor NADPH. So if we keep in mind some of these points, I think that is enough. So actually we don't need to learn each of the step here. Just what particular event occurs in this pathway, where it occurs, what is the name of the enzyme or protein, and also what is the provider of the electrons. Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay, so by that, I think this is the last slide of lipid biosynthesis. If you have any question, you can ask me about this lipid. As I said that our quiz number 5 will be on carbohydrate biosynthesis the lipid biosynthesis, that is the last lecture. I think we have covered. So if you don't have any, then I will start the next one. Okay, can you see it?
Yes, ma'am.
Yes, ma'am.
Okay, so the next lecture is about the biosynthesis of amino acids, but here another one we are seeing heme. We will briefly focus on that. So the first one is about the nitrogen cycle. I think we have also discussed this cycle at the beginning of the metabolism introduction, during the introduction of metabolism, if you remember. So we know that the living organisms, they get nitrogen from the atmosphere, and there are some nitrogen fixing bacteria and archaea which are able to fix the nitrogen as ammonia, and then this ammonia is actually oxidized into nitrite or nitrate, eventually nitrate, and that process is known as nitrification. And there are again plants or other microbes which can actually accommodate this ammonia into amino acids or other nitrogen containing compounds, and those are the like reduced form of nitrogen. And when the nitrogen is oxidized, we get the oxidized form of nitrogen. Those are nitrite and nitrate. And eventually these oxidized form of the nitrate, they actually undergo denitrification by some again denitrifying bacteria, archaea and fungi, and by that the nitrogen will be released into the environment. And in between these, there is another group of bacteria which are called anomox bacteria. Anomox bacteria, they are actually present in an aerobic environment, and they also can release the nitrogen from ammonia or nitric oxide. So that is the nitrogen cycle. Next one, we will focus on this anomox group of bacteria, where actually the nitrogen cycle is short circuited. And these bacteria, they are an aerobic bacteria, and they actually do the anaerobic ammonia oxidation, and by which the ammonia and the nitrite is converted to nitrogen. And there it is about 50 to 70% of this ammonia to nitrogen conversion happening in the biosphere, that is done by this particular pathway. And the reactions are here. The description is in this slide, but if you look at the reaction, that is the anomox bacterial reaction, and their membrane is called anomoxome membrane. Within that membrane, the pathway is occurring. If you look at this, the cytosol, this is the membrane, which is also two layers, and within the membrane all the enzymes are present. So at first, if you look at this, in the cytosol of the bacteria, there are the ammonia and the ammonium hydroxide. They undergoes the reaction which is called hydrazine hydrolase enzyme, doing the hydrolysis, and that we get hydrazine N2H2, sorry, N2H4, that happening in the anomosome, and water is released, and this hydrazine then undergo oxidizing reaction by the enzyme hydrazine oxidizing enzyme, where we can see the release of the nitrogen. So nitrogen is released, but at the same time protons are produced. So those protons are actually transported to the cytosol of this group of bacteria by a proton pump. And again, the proton pump, like we have seen in case of mitochondria, the proton pump is associated with ATP synthesis. When the proton is transported into the cytosol, the ATP synthesis then synthesize ATP from ADP and inorganic phosphate. Okay, now when the protons are released, that also causes the release of electrons. So those electrons are utilized by the enzyme, nitrite reducing enzyme, which actually reduce the nitrate oxide, and it gives rise to the formation of ammonium hydroxide. Okay, so that is the pathway, very short pathway, but that is also actually causing the release of the nitrogen, as well as like by which eventually it is the energy which is generated that is utilized to synthesize ATP. Is it clear? Hello, is it clear?
Yes, ma'am.
Okay, next one. So the normal nitrogen fixation, which we have actually seen at the very first slide, those are done by some bacteria, archaea. So they have the ability to fix the atmospheric nitrogen. And particularly the certain bacteria are called cyanobacteria. They are present in the soil as well as they are present in fresh and salt waters. Other group of bacteria, they are called methanogenic bacteria because they are anaerobic bacteria and they get their energy from carbon, energy and carbon, that is the carbon is required for living organism, and they get it from hydrogen and carbon dioxide, and by that they produce methane. Okay, there are other bacteria as well. Those are mainly soil bacteria. Examples are Azobacter species, and then there are nitrogen fixing bacteria, which actually live as the symbionts in the root nodules of leguminous plants. Okay, some plants they have noodles like structure in their roots, and within those noodles there are nitrogen fixing bacteria. So actually those plants are very rich in protein. Okay, now the very first step, if we go back to the cycle here, if you look at this, where the nitrogen is fixed as ammonia. Okay, ammonia, and then that is eventually converted into soluble forms of the nitrogen. Those are the nitric oxides such as nitrites, nitrates, and eventually amino acids. Okay, now this fixation, like the carbon dioxide fixation, we have learned in the during the biosynthesis of carbohydrate. So here, these organisms, they are fixing the atmospheric nitrogen, and this fixation is done by a protein complex that is called nitrogenous complex. Okay, nitrogenous complex is a protein complex and it is fixing the atmospheric nitrogen. And this complex is made up of two components. One is called dinitrogenous reductase, and another one simply the dinitrogenase. Is it clear? Nitrogen fixation occurred by a protein complex which is called nitrogenous complex that is made up of two components. One is dinitrogenous reductase and another one is simply dinitrogenase. Okay, so now we look at the reaction or the fixation of nitrogen by this nitrogenous complex. That actually all those microbes we have mentioned, they have this nitrogenous complex or protein complex. Okay, now in this nitrogen fixation reaction, electrons are required. And if you look at the reaction, actually, the electrons or the source of electrons is from the reaction where pyruvate is converted to acetyl CoA. So we have already learned this reaction elaborately done by the enzyme pyruvate dehydrogenase complex. Okay, during that reaction, electrons are released. Those electrons are actually, that means ultimately it will be given to the final component of the complex, which is the dinitrogenase. But that is transported by two electron carrier. One is ferredoxin or flavodoxin. Okay, there are one of the two options, ferredoxin or flavodoxin, and from there it will be transported to dinitrogenous reductase. So here if you look at this, so we get the electrons from the reaction of pyruvate to acetyl CoA that is given to flavodoxin or ferredoxin. Then that goes to the dinitrogenous reductase and dinitrogenous reductase actually needs to be activated, and that is done by ATP, participation of ATP, and now it is activated. That means it is now able to transfer the electrons to the another compound, that is the dinitrogenase of the complex. So this dinitrogenous reductase, it has ability to reduce the dinitrogenase by 1 electron at a time. Okay, so it can actually deliver a single electron to dinitrogenase. But to fix the nitrogen, that means one molecule of nitrogen is fixation requires 6 electrons. Okay, so that means sequentially one electrons is like given or transported, and ultimately altogether 6 electrons are required to fix one molecule of nitrogen. Okay, now at the end, because when the nitrogen is fixed, actually nitrogen is reduced and the electrons are given, and additional 2 electrons are required, because when electrons are like given to its substrate, protons are also added. We learned that. So here at the same time 2 protons are reduced to hydrogen. So altogether we actually get like ammonium hydroxide, ammonium ion, and also the hydrogen ion. That is the result of the nitrogen fixation. Okay, but altogether total 8 electrons are utilized to fix one molecule of nitrogen. Is it clear?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
This reaction, this pathway is happening in all the microbes which has the ability to fix the nitrogen. Only the previous one, Anomox reaction, there are the certain bacteria, Anomox bacteria, they can do the other pathway. But other groups of bacteria, they are doing this reaction. Okay, now we get ammonia. So what will happen to the ammonia? Ammonia ultimately that will be utilized by the microbes specifically, or we can say by the plants. They will be incorporated into amino acids or other nitrogen containing biomolecules. And during this assimilation reaction of ammonia into amino acids or nitrogen containing biomolecules, 2 amino acids are actually functioning, and they are glutamate and glutamine. If you remember, during the catabolism of amino acids, we learned that the 2 amino acids, glutamate and glutamine, they are actually functioning in the collection of amino group from all the different tissues of our body. And here, in return, or in a reverse direction, these 2 amino acids, they are providing the amino group to other amino acids and other nitrogen containing biomolecules. Among these 2, glutamate is the one which is actually providing the amino groups to the other amino acids, and that happens by the reaction, transamination reaction. We already learned that reaction, transamination reaction, if you remember. And in case of glutamine, glutamine has the amide nitrogen, so that is the source of the amino group, and that amide nitrogen is give the amino group in a different biosynthetic processes. So we can say altogether there are 2 reactions by which the amino group is assimilated, and one of the reaction is catalyzed by the glutamine synthetase, Actually, that reaction we have learned before, where glutamate interacts with the amino group and give rise to glutamine. And this reaction occurs in mammals. And second reaction, the glutamate is produced from glutamine, and that reaction is catalyzed by the enzyme glutamate synthase, and that occurs in plants and bacteria. So here are the two reactions. The first reaction we have learned several times, if you remember, during the function of ATP, how ATP actually participates in a reaction at that time, as well as during the catabolism of amino acid. So here, at first, glutamate interacts with ATP. Actually, ATP provides the phosphoryl group to the glutamate, and we get gamma glutamyl phosphate and ADP. And in the next step, the gamma glutamyl phosphate interacts with amino group. So actually, the phosphate group is replaced with the amino group, and we get glutamine. So if you add these two reactions, you will get this single step reaction. Okay. And the second enzyme, glutamate synthase, which catalyzes the reaction of alpha ketoglutarate, glutamine, NADPH, and proton. So we get two molecules of glutamate, because the gamma amino group is actually transported to the keto group of the alpha ketoglutarate, which ultimately becomes glutamate. And even glutamine, after releasing the gamma amino group, that becomes glutamate. So in the end result, we get two molecules of glutamate. Okay, next reaction, sorry. Now if we add these two reactions, though they are like catalyzed by two different enzymes, but the net reaction will be alpha ketoglutarate, ammonia, NADPH, ATP, and we get glutamate. Okay. There is another pathway where that is also actually a minor pathway catalyzed by L-glutamate dehydrogenase, and that occurs in all the organisms. And in that reaction, alpha ketoglutarate, ammonia, it interacts with ammonia, and electrons are provided by NADPH, and we get glutamate. Okay. So these are the reactions by which the ammonia is incorporated into biomolecules, specifically we can say in case of amino acids. Okay, now we look at some of the type of reactions which are actually important reactions in the biosynthesis of amino acids as well as nucleotides. Number one is the transamination reaction, as well as some rearrangement reactions. And these reactions, transamination, because during the catabolism, as well as we have just seen during the amino group assimilation, these reactions are very important. And these reactions, the enzymes which are catalyzing these reactions, they have a prosthetic group, and that is pyridoxal phosphate. Then another type of reaction, which are like transfer of one carbon groups. Okay, one carbon groups are transferred in between two substrates, and that actually, again, the enzymes which are doing these reactions, they need cofactor. And if the cofactor is tetrahydrofolate, they usually transfer the aldehyde group or the alcohol group, CHO or CH2OH. If the cofactor is N-adenosyl methionine, then the transfer occurs at the methyl group. Okay, that is the transfer of one carbon groups. And the third one is transfer of the amino groups derived from the amide nitrogen of glutamine. Okay, amide nitrogen of glutamine, and that enzyme is called, the enzyme called is glutamine amidotransferase. Okay, because the nitrogen is the amide nitrogen, glutamine amidotransferase. Okay, now we will, because we already learned about transamination reaction, And transfer of one carbon group, we are not going to focus here, but that will come during the biosynthesis of nucleotides. But the third one we are going to learn here. So in the next slide, this is about the reaction catalyzed by the glutamine amidotransferase. And if you look at the figure, so this is the enzyme, and you can see the enzyme has two domains. The first domain is the glutamine binding domain, and another domain is the amino acceptor domain. So we can say this enzyme has two active site where two different substrates are interacting, and one is interacting with glutamine, another one is interacting with amino acceptor domain. And these two domains, they are connected by a channel, and that is called ammonia channel. Okay, now there is a specificity of the glutamine containing binding domain. There is a conserved element, and that is what a cysteine residue is present at the active site of this glutamine binding domain. So if you look at the structure, the cysteine residue, which has the sulfhydryl group SH, and here is the glutamine amino acid interacting with the active site. So we already know that the sulfhydryl group is actually the nucleophile, so it attacks the carbonyl carbon here, and because of that, that causes the release of the amino group or release of amide ammonia. Okay, and that is released. So that means when it is released, it will travel through the ammonia channel and goes to the amino acceptor domain, where the amino acceptor domain is already attached or bound to the amino acceptor, and the amino acceptor, they can be different types. So two types of amino acceptor is having keto group. So usually the acceptor molecule need to be activated. Again, the activation occurs by the participation or phosphoryl group transfer of ATP. So once they are activated by the phosphoryl group of ATP, then they are like able to bind with the amino group. So depending on the what type of the acceptor molecule, so we will get the final product. So here if you look at this, water is actually required in this reaction. So if it is uh, the hydroxyl group, then we will get the first one RNH2, and another compound will be released. Or if it is a keto group, so if you look at the keto, usually the keto group is not keto, we can say keto group is not activated, or it does not require the activation by ATP, so it is ready to accept the amino group. So in that case, we will get the water. The product byproduct is water. So this is the reaction by which the amide nitrogen of glutamine is transferred to an acceptor molecule by the enzyme glutamine amidotransferase. So obviously this will happen after the assimilation of amino group by the amino acid here. In this case, the amino acid is glutamine. Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay. Okay, now actually the biosynthesis of amino acids, and we know that there are 20 amino acids. Actually, all of these amino acids, they are synthesized from the intermediates of glycolysis, citric acid cycle, or pentose phosphate pathway. And the nitrogen, because the amino group is present in all the amino acids, So this nitrogen will enter by participation of glutamate and glutamine, because they are the donor of the amino group. So this is the flow chart. If you look at this, so we already learned about glycolysis pathway and what are the products here or intermediates of the glycolysis pathway. The 3-phosphoglycerate, if you remember, 3-phosphoglycerate, actually that can be used to synthesize the 4 carbon sugar erythrose 4-phosphate, okay, or here phosphenol pyruvate, that directly can be utilized to synthesize tryptophan, phenylalanine, tyrosine, or the pentose phosphate pathway, we get the ribose sugar. From the ribose sugar, we get histidine, or from the 3-phosphoglycerate, serine is synthesized. Actually, that pathway we have learned from 3-phosphoglycerate to serine in the carbon dioxide fixation. And from serine, we can get glycine as well as cysteine, and then pyruvate. From pyruvate, we can get alanine. So actually, we learned this pyruvate to alanine that happens by transamination reaction, as well as other amino acids are synthesized from pyruvate. Or the intermediates of TCA cycle, if you look at here, specifically oxaloacetate, and we already learned oxaloacetate undergoes transamination reaction, we get aspartate, and then from aspartate we get asparagine, methionine, threonine, lysine, or from alpha ketoglutarate we can get glutamate. So that is also like transamination reaction. So glutamate is providing the undergoing transamination, and we get glutamate, and from glutamate we get glutamine, proline, and arginine. Okay, so again, like before, we actually did not discussed each of the 20 catabolic pathways of the 20 amino acids. Here as well, we are not going to discuss about the biosynthesis pathway of all the 20 amino acids. Just we are seeing what are the precursors from where these amino acids are synthesized. So this is the table which is actually showing the particular metabolic precursors of the 20 amino acids. Okay, in the amino acid biosynthesis pathway as well as nucleotide biosynthesis pathway, there is an intermediate. We called it the notable intermediate, and that is 5-phosphoribosyl 1-pyrophosphate. So if you look at the structure, the pentose sugar, it has a phosphate group at carbon number 5, and 2 phosphate groups attached to the carbon number 1. That is the pyrophosphate. Okay, the 2 carbon phosphate groups, that is the pyrophosphate, and in number 1, 5 carbon is the phosphate group. Okay, so this is abbreviation is PRPP, and full name is 5-phosphoribosyl 1-pyrophosphate. And what are the precursors of this one? Precursors are ribose 5-phosphate, and we know from where we get the ribose 5-phosphate. Can anyone tell me? How does the cell synthesize ribose 5-phosphate?
Pentose sugar pathway.
Yes. So from pentose phosphate pathway, we get ribose 5-phosphate, and then in this reaction, ATP is participating. So ATP is providing the pyrophosphate, which is attached to the number 1 carbon of the ribose sugar, and give rise to 5-phosphoribosyl 1-pyrophosphate, and ATP is becoming AMP. And the enzyme here, according to the name of the product, the name of the product. So here the enzyme is ribose phosphate pyrophosphokinase. That is the enzyme which is catalyzing this reaction. And this is very important intermediates which is used in biosynthesis of amino acids as well as nucleotides. Okay, now actually we are going to briefly look at the biosynthesis pathway of aromatic amino acids. And we know that we humans or mammals are unable to synthesize aromatic amino acids. They only occur in bacteria, fungi, and plants because they are the essential one, and they have the ring structure. And there are three aromatic amino acids: tryptophan, phenylalanine, and tyrosine. Okay, so actually they have ring structure, but the precursors of these amino acids are the simple aliphatic molecules. So that means the simple aliphatic molecules eventually they become circular one, and wherever the double bonds are required, the double bonds are formed, and we get the aromatic amino acids. Okay, what are the simple precursors here? The simple precursors in this pathway, one is erythrose 4-phosphate, that is the 4-carbon sugar, erythrose 4-phosphate, and another one, phosphoenol pyruvate. So erythrose 4-phosphate has 4 carbons and phosphoenol pyruvate has 3 carbons. So when these two precursors, and they are aliphatic, two precursors, sorry, erythrose is the circular one, they combine and they give rise to an intermediate that is called shikimate. Okay, shikimate. So actually shikimate is the intermediate in this biosynthesis pathway, and shikimate is made up of 7 carbons, and the 7 carbons are coming from erythrose 4-phosphate and phosphoenol pyruvate. Okay, now another important intermediate in the biosynthesis of aromatic amino acid is chorismate. Chorismate actually happens in 3 steps. So the first step where we get shikimate, that actually occurs in 4 steps, and then from shikimate to chorismate it occurs in 3 steps. And here the chorismate, it has additional 3 carbons, and these additional 3 carbons are provided by another molecule of phosphoenol pyruvate. And actually chorismate is the one, that is the one or common intermediate which ultimately lead to the biosynthesis of these 3 aromatic amino acid. One is going to the tryptophan, and another one which ultimately leads to the biosynthesis of phenylalanine and tyrosine. Okay, now here, because we are not learning elaborately each of the steps of this biosynthesis, we are just learning the precursors and the important intermediates. So for this, for this course at that moment, that is enough for us to keep in mind. We don't need to learn each of the steps. Even in the next slide, this is the reaction, but I am not encouraging you or telling you you need to learn this reaction. You don't need to learn these reactions. Is it clear what you have to keep in mind? The precursors,
Yes,
the
ma'am.
important intermediates, that all. We don't need to learn the each of the steps of the reaction as well as the enzyme. No need. Okay, next one, histidine biosynthesis. This is also essential amino acid. That means we cannot synthesize it. So we have to make sure this is coming from our diet. And histidine has 3 precursors. So here you can see the involvement of that PRPP, which we have learned, that is very important intermediate. So from PRPP, it is providing 5 carbons, because PRPP in its structure, it has the ribose sugar. From that ribose sugar, 5 carbons are coming. Then there is ATP as well, the purine base, the purine ring structure of ATP, that is providing a nitrogen and a carbon. And then the participation of the amino acid glutamine, which is actually providing second ring nitrogen. So that is the distribution of all the atoms of the histidine. Okay, now like the previous one, here as well, we will just focus on the important steps. The very first step where the ATP and PRPP. Okay, these two precursors, they condense together, and the condensation occurs in between the carbon nitrogen 1 of the purine ring of ATP and the carbon 1 of the ribose sugar of PRPP. So that means the ring structure of the purine that is opened, and that will actually linked to the carbon 2 of the adenine of the ribose. So these two are linked by this condensation reaction. And in the next step, the ring structure of the histidine, which is called the imidazole ring, that is known as the imidazole ring, is formed, and during that reaction another nitrogen is required, and that nitrogen is donated by the glutamine. Okay, so that these two are the major steps in the histidine biosynthesis pathway. So as well as here we have to learn what are the precursors and what are the major steps. Again, we don't need to focus on the steps, individual steps in the biosynthesis as well as the enzymes. But important thing in this biosynthesis is that where we can see ATP. Okay, ATP is one of the metabolite here, not the source of energy. Okay, but up to now, before this one, we have not seen such kind of reaction. Like always, ATP is providing energy by transferring the phosphoryl group, pyrophosphate, or adenyl group. But in this reaction, ATP is participating as a metabolite. So it seems that the major function of ATP we learned, that is the it is providing energy. But here we are seeing ATP is participating in a reaction. So it seems that it is a waste of energy for the cell. So actually, ultimately, that is not happening, because the intermediates which are produced in this pathway that lead to the purine biosynthesis pathway. So here is the reaction. Again, it is not necessary to learn the chemical structure. So if you look at the reaction, actually, where we said that PRPP and ATP is participating in the reaction, and after the reaction, actually, we get two intermediates. Okay, so one of the intermediate which is containing the imidazole ring structure, that will go to the histidine pathway, but other intermediate that actually leads to the purine biosynthesis pathway, and ATP is the purine. ATP is the purine. So for cell, it is not like waste of energy, that is the ATP. So eventually another molecule of ATP is synthesized by this pathway. So if we can learn that, that is enough for us at this stage. So we don't need to memorize all of them, but according to the key steps, you can see PRPP, ATP, and then another nitrogen is coming from glutamine, and we get the imidazole ring, and that leads to the histidine biosynthesis pathway. Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay, so actually, we can say, because rest of the other pathways, if we go back to that flow chart, if you look at here, some of them are actually very simple reactions. They just synthesize by simple transamination reaction, and those we have actually learned during the catabolism of amino acids as well. So from this, you can clearly figure it out how the reactions are happening. Okay, so if you look at this, like just we learned about this histidine. So ribose 5-phosphate is the one of the precursor actually, and 3-phosphoglycerate to serine. Actually, we learned this. If you go back to that carbon dioxide fixation, then we have learned it. And as well as here, that is the other one that we just focus the important uh, steps of the aromatic amino acids. And pyruvate to alanine, that is the simple transamination reaction, pyruvate to alanine. Oxaloacetate to aspartate, simple transamination reaction. Alpha ketoglutarate to glutamate. So all these reactions we have already learned actually. So some are like further steps are required to synthesize other amino acids. So we just focus on the two important or complex pathways, which are aromatic amino acids and the histidine. Okay, so amino acids we already learned, they are the building blocks of proteins. Actually, amino acids not only functioning as the building blocks of proteins, amino acids are also precursors of many important biomolecules. And the list are here. Some of you already know: hormones, coenzymes, nucleotides, alkaloids, cell wall polymers, porphyrins, antibiotics, pigments, and neurotransmitters. So actually, if we look at this, like hormones, then antibiotics, neurotransmitters, they are actually protein in nature. Okay, majority of them are protein in nature. Whereas other compounds we can see here, coenzymes, nucleotides, alkaloids, cell wall polymers, porphyrins, pigments. So they have like complex structures. So in their structure, they have the amino acids. So our body, even like if you remember during the protein macromolecule, we learned that majority of our body components or parts are made up of proteins, and proteins have numerous functions. Okay, they are performing numerous functions in our body or for the living organism. We are just focusing here about the biosynthesis of porphyrin. If you remember, porphyrin is the part of the heme prosthetic group, and that is present in the protein, hemoglobin, and cytochrome. So porphyrin has a ring structure, and that is called the porphyrin ring or porphyrin nucleus. And the precursor or the molecules from where this porphyrin is synthesized, that is called the porphobilinogen. Porphobilinogen, that immediate intermediate of the porphyrin. And porphobilinogen, that comes from another intermediate or metabolite, that is called aminolevulinate. Okay, now we can say this is the common intermediate, of porphyrin, that is aminolevulinate. But because of the differences in the organisms, there are two pathways to synthesize aminolevulinate or deaminolevulinate. And the differentiation is that higher eukaryotes, they synthesize the aminolevulinate from different precursors than the others. So here the simple precursor of deaminolevulinate is the amino acid glycine. Glycine is the amino acid, and that is very simple amino acid. And another precursor, that is the succinyl CoA, which is 4 carbon compound. So these two, they give rise to the metabolite, which is called alpha amino beta ketoadipate, and ultimately that is decarboxylated to 6-aminolevulinate. So the major steps are just mentioned here. In case of plants, algae, and bacteria, that has different precursors and different pathway, and they produce 8 amino levulinates, and their precursors are glutamate. Okay, glutamate is the amino acid, so that is the precursor of amino levulinate in plants, algae, and bacteria. So this glutamate, at first it is esterified by the tRNA, and then it is reduced by NADPH into glutamate, into glutamate 1 semialdehyde, and then the tRNA is released, and in the second step, by the transamination reaction, glutamate 1 semialdehyde is converted to 6 amino levulinate. So that is the pathway here, which is totally different from the pathway happening in higher eukaryotes, or particularly we can say in mammals. So we get the common intermediate of this pathway, which is 6 amino levulinate. Now, two molecules of this 6 amino levulinate, they condense together to give rise to the immediate intermediate or immediate precursor of porphyrin, that is porphobilinogen. This porphobilinogen undergoes many enzymatic steps, and where ultimately 4 molecules of porphobilinogen, they give rise to protoporphyrin. Okay, so protoporphyrin is actually ultimately it has the porphyrin ring structure. And the next step, because we know that the ring structure or the prosthetic group, which is called the heme, it has a iron atom, an iron atom. So the next step, the iron atom is incorporated into this protoporphyrin ring, and that is done by the enzyme ferrochelatase. And finally, we get the porphyrin ring, which is the heme prosthetic group of the protein, hemoglobin, and cytochrome. So here, the points we have to keep in mind: the precursors and some major steps occurring during the biosynthesis of porphyrin. Is it clear?
Yes, ma'am.
Okay, so this next slide is actually it is shown for the mammals, so I think this is not so important for us. I think that is up to this one. It is okay. Now, if you have any question, you can ask me. Okay. Yes, if you have any question. No question? No question. সব বুঝে গিয়েছি। হুম?
মিস পড়তে হবে, না হলে তো বুঝতে পারছি না যে কোথায় সমস্যা।
হ্যাঁ, পড়তে তো হবেই। আরো দ্রুত পড়া উচিত। তোমাদের তো 19 তারিখে পরীক্ষা না?
জি মিস।
Your exam is on 19th. So আমরা যদি শনিবারে after the exam, I think the next lecture is also very short one. We can complete that lecture. Okay?
ওকে মিস।
Okay, so Mr. Talha, you have question?
ম্যাডাম
ওয়ালাইকুম আসসালাম।
শনিবার দিন ম্যাডাম ক্লাস টাইমে মানে 11:50 তে আমাদের প্রেজেন্টেশন আছে, মানে কুইজের পর, মানে কুইজ টাইমটার সময়, তখন কি ক্লাস হবে সেদিন 11:50 আমাদের?
ক্লাসটা হয়ে গেলে কি ভালো না? না হলে তো, কারণ that another lecture 19 number lecture that is actually a little bit it has more content than other one. So আমরা যত দ্রুত শেষ করব তত তো তোমরা ধরো পড়ার টাইম পাবা, তাই না?
জি ম্যাম, কিন্তু ম্যাডাম সেদিনই আমাদের 11:50 তৌফিক স্যারের 2-3 একটা প্রেজেন্টেশন আছে। আমরা লাইক কুইজটা শেষ করে মানে ডিরেক্টলি সেই প্রেজেন্টেশনে চলে যেতে যাচ্ছিলাম।
কয়জনের আছে?
ম্যাডাম 2 জনের এরকম।
সো 2 জন আছে, 2 জন চলে যাবা। এখন তো আর কিছু এখন আর কিছু করতে পারবো না।
জি
সো তোমরা আমি সেদিন ইনশাআল্লাহ টাইমলি চলে আসবো। সো ইফ you should also come in time so that we can start on time. Once the quiz is over, you can go.
জি ম্যাম, থ্যাংক ইউ
তো
ম্যাম।
আমরা যদি লেকচারটা শেষ করতে পারি তাহলে পরের লেকচারটা আমরা মানে ধরো একটু টাইম নিয়ে শেষ করা যাবে এবং at the same time you will get time to prepare for the exam.
জি ম্যাম, থ্যাংক ইউ ম্যাম।
ওকে, আমি এর আগে দিন বলছিলাম যে আমাদের কোশ্চেন প্যাটার্ন, তাই না?
ইয়েস ম্যাম।
ওকে, কোশ্চেনের যেটা আমাদের ফাইনালটা শুরু হচ্ছে লেকচার 11 থেকে, তাই না?
জি ম্যাম।
ওকে, সো অ্যাকচুয়ালি যদি আমরা সেভাবে দেখি, বিকজ আমরা তো অ্যারোবিক রেসপিরেশন অলরেডি শেষ করেছি মিডে। ওকে, অ্যান্ড দ্য next one, which is the TCA cycle and oxidative phosphorylation, these two are common for all the nutrients metabolism, তাই না? Whether it is glucose, fatty acid and amino acid. So আমি যদি সেভাবে চিন্তা করি, ওকে, সো যদি আমি সেটা চিন্তা করি তাহলে ধরো ফ্যাটি অ্যাসিড ক্যাটাবলিজম থেকে একটা, তারপরে অ্যামাইনো অ্যাসিড ক্যাটাবলিজম থেকে একটা, আর টিসিএ সাইকেল অ্যান্ড অক্সিডেটিভ ফসফোরাইলেশন, এই দুটোকে কম্বাইন করে আমি একটা কোশ্চেন চিন্তা করছি। ওকে, কারণ টিসিএ সাইকেলের পরেই অক্সিডেটিভ ফসফোরাইলেশন, তাই না? মেটাবলিজমের যে ক্যাটাবলিজমের যে সিকোয়েন্সটা আমরা শিখেছি। ইজ ইট ক্লিয়ার? তাহলে এখানে যদি আমরা তিনটা হলো, তারপরে এখন রেস্ট অফ দ্য লেকচারের দিকে যদি তাকাই, বায়োসিনথেসিস। সো এখানে যদি আমি বলি ফ্যাটি গ্লুকোজ, লিপিড, মানে কার্বোহাইড্রেটে একটা, লিপিডে একটা, দুটো চলে গেল, তাই না? তাহলে হয়ে গেল কয়টা? 5টা। আর এখন হচ্ছে আমার 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 এই তিনটার মাঝে অপশনাল হয়তোবা থাকবে। ওকে, সো 17, 18 and 19 হয় এখানে এমন হতে পারে 17 এর সাথে 19 অথবা 18 এর সাথে 19, এরকম কিছু একটা হতে পারে। বোঝা গিয়েছে?
জি ম্যাম।
ইয়েস ম্যাম।
সো আপাতত আমি মানে এইভাবে চিন্তা করছি, কারণ আমরা বেশি ফোকাস করেছি ক্যাটাবলিজম, আর বায়োসিনথেসিসের ক্ষেত্রে আমরা মেইনলি ফোকাস করেছি অ্যাকচুয়ালি কার্বোহাইড্রেট, তারপরে লিপিড, ওকে, আর অ্যামাইনো অ্যাসিড আর যেটা নিউক্লিওটাইড আমরা অনেক ব্রিফলি ফোকাস করেছি। বাট 19 যেটা ইন্ট্রিকেশন অফ মেটাবলিজম আমরা বলতে চাচ্ছি, অ্যাকচুয়ালি দ্যাট ইজ ভেরি ইম্পর্টেন্ট। মানে আমাদের ফিজিওলজির জন্য দ্যাট ইজ ভেরি ইম্পর্টেন্ট। That will also help you for 2 or 3 course, GB 2 or 3 course. So এটা হচ্ছে আমার outline for the final exam question pattern. So now if you have any question, you can ask me regarding that. কোনো কোশ্চেন আছে? তোমাদের পপ কুইজের রেজাল্ট তো দিয়ে দিয়েছিলাম না? লাস্ট
জি
জি
ম্যাম।
ম্যাম।
কোনো কোশ্চেন আছে ওটা নিয়ে?
নো ম্যাম।
কি ইসের কি কোশ্চেন ছিল? শর্ট কোশ্চেনে কি কোশ্চেন ছিল? একটাই তো ছিল বোধহয়, না?
জি ম্যাম।
কি ছিল সেটা কোশ্চেনটা? ভুলে গেছো? ফর্ম অ্যাক্সেস করতে পারো না তোমরা? আফটার রিলিজিং দ্য স্কোর you should be able to
হ্যাঁ ma'am, পারি। কোশ্চেনটা এরকম ছিল, হোয়াট আর ডিফারেন্ট টাইপস অফ ইলেকট্রন ট্রান্সফার অকার ইন অক্সিডেটিভ ফসফোরাইলেশন?
হুম, তো মেজরিটি কি আনসার দিছো বলো? হুম
3 টাইপস।
থ্রি টাইপস কি ছিল সেটা? যার অ্যান্সার কারেক্ট হয়েছে সে অ্যান্সার করো। থ্রি টাইপস কি কি? হোয়াট আর দ্য থ্রি টাইপস? হ্যাঁ,
ম্যাম হাইড্রাইড আয়ন আর একটা ম্যাম ভুলে গেছি, আমার এটা হয় নাই পুরোটা।
কি ছিল? ফেরাস টু ফেরিক নাকি? তারপরে ছিল হাইড্রাইড আয়ন। আরেকটা কি ছিল?
হাইড্রোজেন।
হাইড্রোজেন। কারণ হাইড্রোজেনের সাথে তো ইলেকট্রন আছে। আর অনেকে কি লিখছো? ওই যে কি সব জন্য লিখছো? অক্সিডেশন, তারপরে কি লিখছো? টিসিএস সাইকেল এরকম বোধহয় লিখছো। আমরা তো শুধু ফোকাস করছি অক্সিডেটিভ ফসফোরাইলেশন আর ইলেকট্রন ট্রান্সপোর্ট চেইন, তাই না? সেখানে কিভাবে ইলেকট্রনস গুলো তাহলে তো কোনো কোশ্চেন নাই। এখন তোমরা বুঝে গেছো কোশ্চেন হবে কেমনে। সো পড়াশোনা শুরু করে দাও তাইলে। তাহলে 19 তারিখে ফাইনাল এক্সাম আর 26 তারিখে প্রেজেন্টেশন, তাই না?
জি ম্যাম।
আর তোমাদের কয়জন আছে? কয়জন আছে? আমরা তো কয়টা গ্রুপ বানিয়েছিলাম?
7টা ম্যাম।
7টা বানিয়েছিলাম। তো কয়জন বেঁচে আছো তোমরা?
ল্যাব এক্সামে দেখলাম অনেকেই হচ্ছে ড্রপ দিয়েছে সেদিন এক্সামের দিন।
সরি, আই ওয়াজ ডিসকানেক্টেড। না, এখন তাহলে যদি ধরো আমরা 7টা গ্রুপ করেছিলাম, কয়জন করেছিল গ্রুপ মেম্বার?
4 জন ম্যাম।
4 জন। তো তোমাদের গ্রুপ মেম্বাররা কি আছে কি না আছে তোমরা জানো না? তারা কি তোমাদেরকে ইনফর্ম করে নাই যে আমি উইথড্র করেছি? হুম
আছে ম্যাম।
সবারই গ্রুপ মেম্বার আছে? কেউ উইথড্র করে নাই? নাকি একটা গ্রুপ ধরে উইথড্র করছে?
ম্যাম গ্রুপ 1 এর উম্মে সালমা মাহি আপু উনি মেবি উইথড্র করেছে।
ওর গ্রুপে কে আছে?
আফরা জাহান তরি আপু উনারা আছেন।
মানে লাইক দেয়ার আর স্টিল 3 মেম্বার্স, এরকম কিছু?
ইয়েস ম্যাম।
আচ্ছা, যদি 3 মেম্বার্স থাকে তাহলে তো আমাদের 3 মেম্বার্স নিয়েই কন্টিনিউ করতে হবে। এমন কি কোনো গ্রুপ আছে যেখানে 5 মেম্বার্স?
ম্যাম গ্রুপ 1 এ 5 মেম্বার্স ছিল।
আচ্ছা, তাহলে গ্রুপ 1 এ যদি 5 থাকে সেখান থেকে তো একজন চলেই গেল নাকি?
জি ম্যাম।
So they are like they still have the 4 members. আমি জাস্ট জানতে চাচ্ছি যে এমন সিচুয়েশন না হয় যে কোনো গ্রুপে ধরো মাত্র একজন and the rest of the 3 members they withdraw the course. এরকম কোনো সিচুয়েশন যদি না হয়, কারো কোনো গ্রুপে যদি এরকম সিচুয়েশন থেকে থাকে প্লিজ লেট মি নো ইন অ্যাডভান্স। ইয়েস, কেন? তুমি টাইমলি সাবমিট করেছিলে না? তাহলে আসেনি কেমনে? হাউ ইজ দ্যাট পসিবল? রিলিজ তো যখন করেছি সবারটা তো একসাথেই করেছি।
ম্যাম আমারটাও আসেনি। আমি আপনাকে মেইল করেছিলাম বাট কোনো রেসপন্স পাইনি।
আচ্ছা, তুমি কেন টাইম মতন সাবমিট করতে পারলা না সেটা তো আমি বুঝলাম না ঠিক। আমি তো 36 মানে তোমাদের যে টাইমটা ধরে দিয়েছিলাম সেটার পরেই তো করেছি।
ম্যাম আমি 34 করে দিয়েছি, কিন্তু সেটা কতক্ষণ যখন 35 হয়েছিল তখন দেখাচ্ছে যে নো লঙ্গার রেসপন্স, বাট আমি 34 সেটা সাবমিট করে দিয়েছিলাম।
তাহলে তো তোমার ইন্টারনেটের প্রবলেম। আচ্ছা, তুমি এর আগে কে যেন বললো, তোমার আইডিটা বলো তো। আনিকা না? আইডি বলো দেখি। না, শুরু থেকে শেষ পর্যন্ত বলতে হবে তো। হুম, 213030। টোয়েন্টি-ওয়ান। তোমারটা তো চেক করেছি, তো তুমি পেলা না কেন? দেখো তো এখন পাও কিনা। ওকে, সো কোনো ভাবে বোধহয় কিছু একটা হয়েছিল, জানি না। ওকে, সো any other question, anything?
আমাদের প্রেজেন্টেশনটা কি ক্লাস টাইমে শুরু হবে?
হ্যাঁ, ক্লাস টাইমই হবে। কারণ 26 এ তো আমাদের ওই যে বলা আছে অ্যাকর্ডিং টু দা ক্যালেন্ডার, আমাদের যে কি যাদের ওই যে 2টার বেশি এক্সাম একদিনে তাদের জন্য আর কি এক্সাম স্লট আছে। সো আমাদের ক্লাস টাইমই করতে হবে।
ওকে ম্যাম।
So please prepare, start the preparation for your exam. Not only this course, obviously you have other courses as well. We don't have time. From the next week you like the exam is starting. So 15th December, from 15th December your exam starts.
মিস
হুম,
17-18 থেকে কি কিছুই আমাদেরকে কমাই দেওয়া যায় না একটু মিস?
17-18 থেকে তো কমিয়ে দিলামই, আর কি কমাবো?
মিস কতগুলা লেকচার মিস, আমাদের অনেক টাফ হয়ে যাবে
তোমার
19 তারিখের মধ্যে।
তুমি আমার লেকচার নিয়ে বলছো কতগুলো? So অনেক ফ্যাকাল্টি they are like taking the exam on the whole syllabus.
মিস এটাই তো আমাদের তো মানে 3-4টা কোর্স, এর মধ্যে সবাই আর কি সবগুলো কমপ্লিট করতে চাচ্ছে, এরপর আমাদের অবস্থা 12টা বেজে যাচ্ছে মিস।
আমার এখানে তো কিছুই করার নেই। So I, like, as a chairperson, I cannot impose any faculty about this. It's their own decision. ওকে, সো এখন একটা মানে আনএক্সপেক্টেড সিচুয়েশনের মধ্যে দিয়ে আমরা যাচ্ছি, আমাদের এখন কিছু করার নাই আর কি। বাট ইনস্ট্রাকশন আমি জানি না অন্য ফ্যাকাল্টিটা কিভাবে পালন করছে। আমাদের কাছে যে ইনস্ট্রাকশনটা আছে সেটাতে বলা হয়েছে যে ডিউরিং দ্যাট ক্লাস টাইম, দ্যাট মিনস দ্য লাস্ট ডে অফ ক্লাস, হোয়াট এভার দ্য কন্টেন্ট ইউ আর এবল টু কমপ্লিট, দ্য এক্সাম উইল বি অন দ্যাট। এটা আমাদের ইনস্ট্রাকশন আছে আর কি। So already the instruction is given to all the faculties of the university. So it is not like the chairperson need to inform them again. Okay, so আমি তোমাদেরকে already তো mention করলাম the question pattern. Now it is your decision. How are you going to prepare yourself? ঠিক আছে, then we'll see you on Saturday 11:50.
আসসালামু আলাইকুম ম্যাম।
ম্যাম আসসালামু আলাইকুম।
ওয়ালাইকুম আসসালাম।
আসসালামু আলাইকুম।
আসসালামু আলাইকুম।